Okay, Lab4. The Lab4 will do an experiment on E. coli cell transformation. Hopefully by this time we'll be able to meet face-to-face and can explain this to you face-to-face, but just in case I'm narrating this lab so you can take a look at it while you want to prepare for this at your convenient time. So I'll post it on the course shell. So transformation of prokaryotic cell like E. coli means you are introducing some genetic elements to the bacterial cells, and normally those genetic elements have some functional genes, so that genetic trait is introduced into the maybe cell, so the cell will maintain the genetic trait or phenotype from the time on. That's what transformation is. And there's another word called transfection. So when you do the similar process in eukaryotic cells, especially animal cells, instead of saying transformation, you call the process transfection. Okay, so transformation, transfection are kind of similar. So transformation of E. coli cells are similar to the transfection of animal cells. Then why do you call transfection instead of transformation when you deal with the animal cells? The reason is transformation is already explaining something else in animal cells. Normally, normal cells becoming cancerous cell, they call that transformation. So, since that word is already taken to explain that cellular process, they decide to choose another word to describe this process. So transformation, transfection, a similar process. So it's change of genetic makeup of cells by introducing new genetic elements. So normally that genetic element could be recombinant plasmid carrying a unique gene. Okay, so that's what transformation or transfection is. So here you go, plasmid plus the functional gene added to it. So normally they don't go in separately. Actually, this is the host cell, so that red part of the plasmid DNA carries the unique gene in there. So this recombinant DNA is moved into the host cell. That's the transformation we're talking about. So this transformed cell now carries that new genetic element, right? So then the question is, how do you make this DNA go into the cell, right? So we'll talk about transformation method. So how do you insert or transfer the genetic element into the cell? These are possible ways of doing the transformation. So one way is you shoot an air gun, okay, with the pellet that's coated with the genetic element. Then those pellets will maybe penetrate through the cell, and by doing so it can deliver that genetic element. So maybe shotgun method, that's what we call it. Another method will be maybe if we have a very fine needle, you can inject the genetic element. You could do that, especially when you deal with animal cells. That's a preferred method of DNA delivery. Another way is you can use a virus, like bacteriophages that can infect bacterial cells. Then by using their natural mechanism, you can deliver viral genome into it. In this case, viral genome contains your gene of interest. Okay, and another method is you can treat the cells with some ions. Okay, so then the ions will weaken the cell wall and membrane structure, and then when you heat shock it, and then DNA can go into the pores that are made through the heat shock treatment. So there are various ways of delivering the foreign DNA element into a host cell. Okay, so this shows a petri dish medium with a bunch of these E. coli cells. So normally this is a selective medium, contains some kind of chemical that will only allow the cells that are transformed to survive, like antibiotics like ampicillin. So non-transformed cells cannot survive. Only the transformed can survive, because the resistance to antibiotic comes from the genes of the element that's just transferred into the cell. So all these cells can survive because they are transformed. Okay, and they may have also different colors, and there's another color-based selection. So white colonies may be the ones that you are looking for, blue ones are not, because color-based difference comes from enzyme synthesis happening in the transformed cell. if the enzyme is still made, gene of interest is not inserted, but enzyme is not made anymore, then the gene that codes for the enzyme, your gene of interest, is added, so the mutation happens, so the enzyme is not made anymore. So the color of the colonies will stay white instead of blue. So actually, among these cells, the white ones are the ones you need. We can talk about that later again. It's color-based selection. Another thing you need to understand is the E. coli cells can be readily transformed. Using various methods, you can transform E. coli cells with the foreign DNA molecule. The DNA can be linear or circular, but circular is a better conformation of DNA when you're trying to transform E. coli cells. One easy, cheap way of doing that is chemical transformation. As I said, the host cells can be chemically treated by adding maybe calcium chloride or rubidium chloride. Those ions can compromise the cell wall, cell membrane structure. Okay, and then the DNA and then the cells are heat shocked at the kind of ambient temperature of about 42 degrees Celsius. Okay, a sudden temperature shift from maybe 0 to 42 degree, they will create transmit a lot of thermal energy coming in. Thermal energy disturbs a lot of things in these. So through the disruption, there may be transient pores made on the cell membrane. Through those pores, your DNA can be transported in, and then cells are recovered from the short heat shock to the optimum condition, growth condition. And then the cells that are not dead through this harsh treatment, and then during that treatment, if cells have taken up the foreign DNA, and they will survive and they'll continue their life, and their lineage, their descendants, will maintain the genetic traits that are developed by the new DNA that came in. And that trait may be ability to survive with the presence of antibiotics, or ability to give no color when there is a color substrate in the medium, things like that. Okay, so that's a chemical transformation method. And electroporation is, like you can zap the cells with high electric voltage, very short electric charge, okay, and like 15 kilo, so 15,000 volts. You can expose the cells to that voltage about very short period of time, maybe 5 microsecond pulse. Okay, and since DNA molecules are charged, negatively charged, they will follow the electric current. Okay, when they do that, some DNA may be trapped inside of a cell during the electro zapping. Okay, so that could be a method of transformation. When you do that, we call it electroporation. Okay, because electric current is making a pore on the cell membrane. That's how you can transfer your DNA into the cell. So we'll talk about this chemically induced transformation method. Okay, so you can grow cells when you do that optimum condition. So cells should be very healthy. Okay, so they should be under the log phase. We'll talk about the cell growth phase again, but normally there are 4 distinctive phases in the cell growth cycle: lag phase, log phase, stationary phase, and death phase. So the log phase is the healthiest cell condition. Okay, they will be actively growing and eating. So you will capture the cells who are very healthy in the log phase, and then you will have to make the cells competent of receiving DNA. How do you do that? By adding some chemicals like calcium chloride, or rubidium chloride. Normally those ions are added. So what do those ions do? Think about your membrane, cell membrane structure. Membrane is a phospholipid bilayer, right? Phosphate head, and then hydrocarbon chain in the middle. Okay, so bilayer is made of 2 layers of phospholipids. So if you look at phospholipid, okay, phosphate head is negatively charged, so there are strong negativity outside of the membrane. Okay, and inside is hydrophobic. So actually your membrane is negatively charged. And if you remember your DNA molecular structure, DNA is also strongly negatively charged because it also has phosphate sugar, that backbone. So PO- is found from your DNA as well as from the plasma membrane of your cells. So negative molecule, okay, is approaching to another negative molecule. What's going to happen? Will they like each other? The answer is no. They will push away each other. So actually, since they are charged the same way, they will not like each other. So to compromise that charge, okay, you want to add calcium ion. So calcium ion is positively charged, right? So if you add a bunch of calcium ion, then calcium will bind on the phospholipid, the surface, and then it also binds with DNA that has few minuses. So calcium ion will neutralize the charge of the cell membrane as well as neutralize the charge of the DNA. That's why you want to add calcium chloride or rubidium chloride into the cells. That's what you do with the cells. We call it making the cells competent. You're basically neutralizing the cell charge by adding those ions. Okay, so that's the first step. And then, yes, I just talked about DNA that having the PO minus charge, so that you want to neutralize that again. So that same calcium and rubidium ion will neutralize the DNA as well. Okay, once that's done, then you're going to heat shock the cells with the DNA. So very brief heat shock, maybe about 30 seconds at a big temperature shift. Okay, maybe 37 degrees, okay, or 42 degrees. Okay, if you expose a cell, all of a sudden there's a flux of thermal energy coming in, and that thermal energy will disturb a lot of structures, including the membrane structure in the cell. Okay, so rapid increase of the thermal energy and it disturbs a lot of cellular structures. That's what heat shock would do. And one of the disruption happening is making a lot of membrane, moving around. While they are moving around, there may be a hole made in the cell membrane. So then through the hole, your DNA will move in to the cells. That's what heat shock would do. Okay, a brief heat shock treatment of neutralized cells and DNA, and that shoves the DNA molecules through the cell because there may be some holes made on the cell membrane. Okay, so that's what heat shock would do. And then recovery, once heat shock is done, that was a harsh treatment. So some cells may die because of it, because they could not recover. But hopefully there may be some cells that can still survive after the harsh treatment. And then you will put the cells back to the optimum growth condition at maybe 37 degrees Celsius with plenty of nutrients and plenty of oxygen. Okay, then some of the cells will recover. They'll come back to life and they'll divide. Okay, so these cells will be recovered and grown continuously, and now at the end you plate the cells, transfer the cells on the medium that has some selective chemicals, maybe some antibiotics or substrate that gives the color and all those things there. So the plating is the last step. So plating is done to select for the cells that have been transformed. So plating is the selection step. Okay. So those are the transformation steps that are important. Okay. So what you will do in this lab, let me talk about. So you'll be given with the frozen competent cells. So cells were treated with the calcium chloride, rubidium chloride. Okay, and they will be kept on ice about 5 to 30 seconds. Okay, and then you will add a certain amount of DNA. So DNA is a plasma DNA that will give you, okay, so a known quantity, about 100 picogram or 1 nanogram of DNA. So you need to know how much you are given with. Okay, and then that DNA will be added to the competent cells. Okay, so since competent cells are added with the calcium chloride, DNA molecules will be also neutralized. Okay, and then once that's done, you'll incubate the mixture on ice for about 30 minutes. Okay, and once that's done, so during the time, hopefully neutralization take place, and then you will heat shock. So from 0 degree to 37 degree, all of a sudden, that's a big temperature shift. We'll do that about 30 seconds. Okay, once that's done, you will keep the cells into the optimal growth medium by adding new fresh medium and moving them to 37 degree shaker. You shake the cells to provide continuously provide oxygen. Okay, and then at the end, they plate the cells on a selective medium, the plate with the antibiotics added there, and you're going to grow the cells overnight. And next day, you'll check the cells if they're surviving or not. So that'll be the entire process. Okay.